Hello, science lovers. Welcome to our Open Minds Teen Science Cafe. We appreciate you coming tonight. We'd like to also greet our online audience. They're tuning in via Facebook Live. Hi. You folks can feel free to ask questions in the comment section when we get to Q&A. Please remember to be respectful of all of our guests, and we remind you to silence your cell phones. We'll be taking pictures during the cafe to post on our social media. Be sure to like, follow, or tag us on Twitter, Facebook, and Instagram.
Thank you, Valerie. So tonight we have a very special guest for you guys. He's been practicing his X-ray vision superpower for several years now as part of his PhD dissertation research and currently as an R&D engineer at Duke University's Shared Materials Instrumental Facility, SMIF, running the micro CT. That was a lot of really big words. I'm proud of myself. His favorite hobby is Instagramming. Anybody else? Yeah? Yeah? No? Okay, whatever. Woo! Yay, Sachi. And his favorite snack foods are baked goods, and his favorite superhero is Gambit from X-Men. Ring any bells, guys? Anyway, so please welcome Justin Gladman. Woo!
Thank you all for coming out. Thank you for the introduction. Today I'm going to talk to you about X-ray vision. So the title of the talk is X-ray vision, science or science fiction. And to be clear, everything I'm going to talk about today is definitely science. The micro CT that we're going to discuss is built on science, and all the researchers that come scan things in our facility are scientists who have very different scientific interests. But sometimes I see the results of these scans from the micro CT, and I'm amazed at how detailed and beautiful they are, and the fact that we can see into things that normally you can't see with the naked eye. And to me, that still seems a little bit magical, a little bit like science fiction. So we'll look at a little bit of both. Superman, of course, has X-ray vision as one of his many, 20 million different superpowers that he has. And you can find advertisements in toy stores and magazines for cheap pairs of plastic glasses that say you can see through walls if you put them on. This is obviously all science fiction, but the theory is not too far off. The use of X-rays are an important part of seeing into objects that we otherwise wouldn't be able to see. And you all know this. You've been to the dentist office or to a hospital visit and had an X-ray. And when they do that, they can see into your body, which you normally can't see with the naked eye. So let's just talk briefly about X-rays. X-rays are like visible light, except they're higher energy, a shorter wavelength, and a higher frequency. And that higher energy means when an X-ray hits you, it doesn't just stop and bounce off. That's basically what visible light does. It hits you or an object in the room, bounces off, and that's how our eye detects all the shapes and colors that are sitting in this room today. But X-rays with this higher energy pass right through you. So your eye can't detect them, but if you put something on the other side of that object, like a piece of film, the film would be exposed and an X-ray image would appear. So this is what this image is on the screen on the right. This is one of the first X-rays taken by the German scientist who kind of discovered what X-rays were. He was tinkering around in his lab and discovered that some of these tubes he was working with were glowing, and he wanted to try to figure out what was going on. He put various things in front and realized that film was exposed and created a picture when he put them in front of these tubes. He didn't know what these rays of light were being produced by this tube, so he just called them X-rays, assuming that they hadn't been described yet. And this was one of the first images he took of his wife's hand, and you can see the bones in her hand, and the ring is that big blob sitting on her finger there. So since then, we've come a long way. In X-rays, we have a lot finer detail, and they're a lot safer. We don't just throw people in front of X-rays for long exposures anymore. We know that they can actually be harmful in too big of quantities. And we can see a lot of detail now using X-ray technology. But there's still a lot left that we can't tell with a simple 2D X-ray. And this visualization kind of gives you an idea of what I'm talking about. On the left there, the pair of hands are folded on top of each other. And you can surely see a lot of amazing detail, the bony structure of each of those hands. But if you wanted to try to tell things about the position of those hands. Is it the left hand on top or the right hand on top? You can't really figure that out from this image unless you really know some subtle anatomy. However, the picture on the right, obviously not hands, this is a little South American monkey called Calamico. And this is a CT scan, so it's not just a simple 2D X-ray, but if I can click it. This is a three-dimensional version of that X-ray. So it's actually thousands of 2D X-rays that are reinterpreted into 3D space. And when we do that, we can talk about the position of the anatomy. Is there a broken bone above or beneath instead of a 2D X-ray where we couldn't necessarily see that detail? So the CT is the next step in this X-ray technology to give us even more information about the things that we're trying to study. So what is a micro CT or a CT in general? You might have heard about this if you've gone to the hospital and had a CAT scan. CT stands for computed tomography. The tomography part is like measuring or mapping out the specimen. And the computed part is the idea that it takes a really strong computer to interpret this really complex data. Basically what it is those simple X-rays that you get at the doctor's or the dentist's office, these 2D flat images, and the computer reads thousands of these at different angles to interpret a 3D shape. So that computation is what makes this unique. This is a medical X-ray on the screen here. It's a big donut-looking thing, and you lay flat on a bed and you roll through it. And in that donut, there's an X-ray source, and on the opposite end, a detector or X-ray film. And it takes thousands of images as it spins around you, getting all that raw data needed to get the final CT image. And I won't go into that complex math because I can't pretend like I actually understand any of it. But the picture on the left gives you an idea of what we're talking about. Those little diagrams with the grids on the left are projections or individual 2D X-rays. And when you do that thousands of times around 360 degrees, around one turn around your specimen, whether it's a human or a Calamico monkey, the computer then can take those slight variations in the angles of the X-rays and reinterpret that as a 3D X-ray image. And on the right there, you see kind of how that looks. The medical CT takes a slice of information from your body, and instead of now being a flat 2D image, it can look at the 3D internal structure of your abdomen. So this is the micro CT that we have at Duke University in our shared materials instrumentation facility. And this is what a lot of people come to use to look at their specimens. It's not a very pretty piece of equipment, but that's because this whole box is lined with lead. Just like when you go to the dentist's office, you get that apron put on you, right? That apron is lined with lead because it protects you from the X-rays that are being zapped at your teeth. Well, same here, and the reason we line it with lead is because X-rays can't penetrate lead. Lead is actually so dense that even though X-rays normally go right through an object, they stop dead in their tracks when they hit this lead surface. So even Superman wouldn't be able to peer into our machine. Luckily, we have a little viewing window up front, so when I'm sitting there, I can check out what's going on. And I'll give you a little peek inside to talk about how this machine is set up. So inside the micro CT, this is just a diagram of what we're about to see. There is an X-ray source, and this is where electrons that are produced hit a piece of metal called tungsten and produce a cone of X-rays. So that yellow cone you can imagine as a cone of X-rays being shot out of that source at some sort of object in the middle of the chamber. And you'll see the object in the next slide is a little USB thumb drive. And then they pass through that object and go to the detector in the back. The detector in this case is just a black screen, and think of it like a digital camera, except this is a digital X-ray camera, and it sends those images directly to our viewing monitors. And this is what it actually looks like inside of our machine there. So you have that X-ray source and target. In the middle, it's a little hard to see. I don't have an arrow to it. You see the USB thumb drive, especially in that bottom right image. And in the very back, that black shiny screen is the detector, which is capturing the image. And when we start up the machine, that cone of X-rays flies back through the USB, And because of that different wavelength, they pass right through and go back and expose the detector. And when those X-rays hit an object, whether it be a USB drive, or in this case, the little diagram is of a human, body, some of those X-rays are transmitted through, meaning they pass straight through. And they'll pass through when the object is not very dense. Maybe just the skin in your body, or in the USB drive, maybe the piece of plastic, they'll go right through. But when it hits something dense, like your bone, or metal pieces in the USB drive, they'll be absorbed and they won't go through. And that difference between the X-rays that are transmitted and absorbed is how we get that image on the other end. So in our little Homer X-ray here, his thick skull is dense enough to stop those X-rays. So that turns into a white part of the image. And then inside his skull, not so much. The X-rays pass right through and expose the film to make it dark. This is what you're used to seeing in a doctor or a dentist's office. Now, some of these images that I have will be a little bit flipped. The dense parts will be black and the white parts will be less dense. That's just because we don't use real film anymore, so it's not a pure negative. So we can make it look however we want, but you'll get the idea of when we're looking at something dense and when we're looking at something that's relatively light. All right, so the last bit of setup is, what is the difference between a medical and a micro CT? I work with a micro CT, but you all are probably familiar with the medical version of it. We already showed an image of what it does. The real difference is the resolution that it can capture. So the image that we get in a micro CT is significantly better and more detailed than a medical CT. These are pictures not from a CT, but from another SEM type of image. And I'm just using these to show you an idea of the scale of what we could see with a micro CT scanner. The picture on the left, you can't see anything at all in this view, because what I have highlighted there is only 5 to 10 microns in diameter. A micron is 1/1000th of a millimeter. So picture a millimeter divided up 1,000 times. One of those segments, 0.001 millimeters, is a micron. So extremely fine detail. And the picture on the right, you have a slightly larger object, about the length of 1 millimeter. And in a medical CT, we could see that, because a medical CT, the resolution that we can see is about 1 millimeter. Anything below that becomes fuzzy and you don't really see it. Anything above that you can image. So in a medical CT, we could kind of get an idea of the second image, but neither image is very good. If these were in a micro CT, though, you would be able to, because of the resolution, see both this. So the red blood cells, the thing that's about 5 to 10 microns, and on the right, the lice is a little bit larger. And we could not only see the lice with the micro CT, but we could see the body segments and fine little hairs on the lice's legs if we wanted to. Now, in reality, we can't scan something like a red blood cell on a micro CT. I'll give you some examples of what we can scan. Again, this is just giving you an idea of the type of scale we're talking about. Another difference is micro CT also means we can only work with small stuff. If we're zooming in quite a bit, we lose the ability to scan a whole human. We can maybe squeeze a little human into the chamber, but we can't capture all the data from its whole body. So the picture on the left, medical CT. On the right, the rubber ducky is about the size of the specimen you would want to throw into the micro CT. Maybe 6 to 10 inches at the very most. And I use rubber ducky here, not on accident. It's not totally random. We gave our micro CT a little Christmas present this year, and it couldn't wait till Christmas, and it does have this superpower, so it decided to peek into its present before it was really allowed to open it to figure out what it got. And so this gift bag is about the size of maybe 10 inches, and that's about the maximum size that you'd want to fit into our machine. And it was pretty excited to find out what it got. Not a rubber ducky, but we are at Duke, and we gave it a blue devil ducky. This is the type of stuff we sell in our bookstore. And so you see in this image, this is a reconstructed visualization of the bag before we ever opened it. You can see the bag at the very beginning there, and we can fade it away and then see the object inside very clearly. All right, so we'll look at a couple different types of data that come out of the CT. There's a lot of cool images that it produces. There's three types that you should keep in mind as we look at some of these images. First off is the projection data. When you think of projection, just think of a normal 2D X-ray that you get at your doctor's or your dentist's office. That's the one where it's just a flat image of your tooth or the bone that you broke to go get fixed. But unlike the doctor here looking at the X-ray of a human chest, we're interested in the small stuff. So I took an X-ray or a projection of a little bumblebee. So that's a little bumblebee sitting in a little plastic tube, and its head is pointed straight up, the rest of its body is down, and you can kind of see that little part arcing out of its head is its little proboscis sticking out, its little tongue sticking out at you. Just like in this image, that bee is using its tongue to lap up the honey. The bee that I collected and I'm scanning here died doing what it loved best, eating honey, and it's sticking its tongue at you now, and you'll see that in the rest of the visualizations. So that's one 2D image, but we actually, like I said, across 360 degrees, take thousands of those 2D images, and you can stream that together into a little video showing how it looks spinning around in our machine. This is the raw data. So even though this is really cool to look at, and you can see a lot of the internal detail, we don't stop here because we still can't look clearly at the internal structures of this bee. So we take it one step further. Again, we have the projection on the left. We do that 1,000 times. And then the computed part of computed tomography reconstructs that data into a 3D X-ray that you can slice up. So on the right side now, that same skull, after it's reconstructed, you can slice through it and look slice by slice through the skull and the brain of this individual. And this is what you see in a medical scanner. For our bee example, you can do the same thing. So now we're traveling slice by slice through that bee from the very bottom to the very top of its head. You'll see it stick its tongue out at you in just a second, right about there. So you see as it goes up from the bottom, you'll see its legs and little tiny fine hairs right here. Imagine how small the hairs on a bumblebee's legs are, and we can see that extremely clearly on this image. So that's the type of detail we're talking about. There in the body cavity, you can see internal detail of the bee, the muscles and the different organs that it has, and then the eyes and the tongue sticking out at you at the very end. So this is the data we're interested, but we can even take this one step further and take that slice by slice image and create a 3D model out of it. We have all the information, so we just generate a surface on it, and now our bee can look like a bee again, spinning around, sticking its tongue out at us. So those are the types of data we'll see, especially those latter two. The slice by slice and this 3D visualization are the coolest ways to look at this data, so I'll try to show you a few more examples. All right, so now on to some applications. And we have so much research going through Duke, and some of these examples are in other institutions. This is just a small slice of all the stuff that I could possibly show you, trying to cover some of the big fields that use the micro CT. The first group that uses it are engineers. The SMIF lab is in our engineering department at Duke, and people in the engineering department use this for characterization and quality control. Characterization means we want to check out our sample without destroying it to see if it is what we meant it to be. We have a big clean room at SMIF, and people are building little microchips for computers. And when they build those microchips, they want to make sure all the little circuits are lined up and attached exactly how they want it to be, but they don't want to destroy it. And to see some of that detail, you'd have to rip pieces off, right? You just spent a long time making it. You don't want to destroy it to see it again. So our micro CT is a non-destructive characterization tool that allows us to inspect this data without messing with the actual specimen. So here's that USB drive again on the left, and you can see we peeled away the plastic all digitally, and now we can inspect all the circuits and wires without ever destroying that device. And the top right is a single projection of, I can't remember if that's a laptop or a phone, but you can see all the circuits and the battery in there. And then just started spinning on the bottom right is another common office items. I may have a guess on what that might be. Remote's a good guess. There's a battery pack on the bottom and a little speaker that disc in the top. So that's a little cordless phone that I have in my office. Say there's something wrong with the phone, but I didn't want to rip the phone apart to see what it was. Now I can check out all the internal circuitry and wiring to see what might be going on in there. But there's a lot of people outside the engineering department that are also interested. And these are people that are interested in collecting and conserving. So building collections of animals and fossils and museum specimens, being able to share them broadly without disturbing or destroying the originals. Right? So we're in a museum right now, and unfortunately, some of the coolest stuff that are in museums are locked away in specimen drawers behind the scenes because they're one-of-a-kind fossils, or they're critters that just we don't have very many of, and we can't risk them deteriorating and being destroyed. Unless you have, funding from an institution or special permission to come visit, which a lot of us don't have that access. So by scanning in the micro-CT and creating these beautiful 3D models, we can then digitize the data and share it broadly. I can post this all online and email it to you all at the end of this talk if I wanted to. Instead of being locked in the museum drawer now, all this stuff is freely available. At Duke, we have a little lemur center next door, so we have a lot of people interested in these primates. So this little Pato, which you see the picture of, we took a scan of it. This guy is a wet specimen, meaning it still has all its flesh and fur on it. We don't want to dissect and destroy it because there's not that many available to researchers. And if one researcher dissects and destroys it, the next researcher can't do anything with it. But we can throw it in the micro-CT because it's non-destructive, digitize it, and then digitally dissect away all that fur and soft tissue to see the skeleton underneath. And when we did that, we quite quickly saw that it has all these little orange nodules all over the place. This is not something we could see on the actual specimen because this is all under the skin. But very quickly with the micro-CT, we open up a whole new line of questioning about what's going on with this individual. Is that little tumors or fatty deposits? Is that something that happens in the wild, or maybe this was a zoo specimen? This is the type of research that can stem off of scans like this. And we actually have an outlet to put that all online. There's a website called Morphosource, which I encourage you to all go check out. This is open to anybody. Teachers, students, the general public. You can get on there and browse through different projects that people are doing, download 3D data, manipulate it, measure it on your own computer. If you have a library or a facility that has a 3D printer, these models are perfect to go and send on to the 3D printer. And you can have Mr. Pato sitting on your desk at home as a 3D printout if you wanted to. There's some really famous specimens like the Homo naledi skeleton, the human ancestor from Africa. When that was published, they also posted all the fossil material on this website. So you can go freely check that out. This democratizes the data. This allows everybody access to some of this really precious data. And hopefully that's going to get people interested in some of these research questions. Okay, so what else? We talked about how fine a detail this thing can see, the micro-CT. So we want to scan really small stuff and see how big and interesting we can make it. Well, about as small as we've done so far is this little ant head. And I actually have the ant head here in this little vial. Most of you probably have trouble just seeing the vial that I'm holding up, let alone the little floating speck in here that is the actual ant head. If you want to come up after and see the real deal, you're welcome to. It's only about 1 millimeter or 2 in length. And the researchers here are interested in this because it's a special ant from, I think, Southeast Asia. And it has these special jaws. It's called a trap jaw ant. These jaws you can see in the pictures of the real animals in the top and the bottom. They open up really wide and slam shut at something like 145 miles an hour. Right? That's 2,000 times quicker than the blink of an eye. And researchers are interested in how they do that. What kind of musculature and setup inside their heads allows them to close their jaws like that? When I see it, though, that's not my first question. My first reaction is that this reminds me of the movie that I grew up watching, Honey, I Shrunk the Kids. I use this example a lot. I realize this movie's starting to become dated. Luckily, with the superhero theme in mind, we have a new comparison that we can make. So this reminds me of Ant-Man now, right? And we can do this. And Ant-Man, the little superhero guy, got really small at the level of the ants, and you could see all this fine amazing detail. What we can do is bring the ants up to the size of us so that we can inspect that same detail on our level. And it's not just that external surface. That picture on the left was a 3D surface that I made, but it preserves all that internal anatomy with the X-rays passing through that ant head. So that all that gray stuff, especially in the back part of the head, that's all musculature. You can see individual muscle fibers running through an ant's head. Think about that. Think about a millimeter ant head, and then a tiniest fraction of that, a single muscle fiber running through its head. Now we can see it and quantify it, and researchers can use that to learn about this species. But we can take this even one step further, and because it's a 3D model, we can send this to the 3D printer, blow it up to a size that we can handle, print it off. The camera here is about to zoom around to the top of it, and you'll see that there's these squiggly lines. The straight lines are just struts that the printer uses to keep everything steady. Those kind of squiggly blobs there, that's the actual musculature that's preserved and now is being 3D printed on a scale that we can see it. So if I were to stop that print right there, you could see the internal musculature of that ant. Instead, I let it finish, and you can see how big that ant is right here, right? So from not being able to see the vial that it was in to pretty clearly seeing the ant head in my hand here, and I could pass this around without really worrying about it being damaged or destroyed, right? So now we have the ability to share this information more broadly. Again, you're welcome to come up and see the 3D print after the fact. Let me put them out here. And just for scale, this is one of the few times in your life that you'll see an ant that's bigger than a mouse. Some other applications. People are interested in the earth sciences. Again, if you have an ability to see through things that otherwise haven't been able to look into, people are interested. So rocks. Rocks, the only way to study them is to crack them open and to look. And the minute you crack them open, you've destroyed your sample. So if you're interested in some rare rock, think about what kind of rocks might be really extremely rare. Rocks that like fall out of the sky, for example. Meteorites. So if you have a meteorite and you don't want to just take a sledgehammer to it, you could throw it in our micro-CT and characterize what's going on inside. And even if it's not a meteorite, these are just different core samples. You can talk about what kind of minerals and deposits and formations are in all these rocks. Also, soil cores. We have a researcher at Duke who's interested in this site in South Carolina, where it's been heavily plowed with agriculture and over time eroded. And now they're trying to grow certain portions of it back. And he's interested in how the soil composition has changed over time. Well, the minute you try to look at soil, you're required to dig it up and disturb that very environment. So what he's done is he's taken core samples, like you've seen probably ice cores or rock cores before, a sample of soil, a foot or two down, fairly undisturbed, put it in the machine, and scan it. And what you see, those dark kind of blue snaky lines that are passing through, those are little roots in the soil. And you can track how that's breaking up and embedded in these different soils from the different regions of this site. And then some of the coolest examples that I think are the coolest examples are archaeology and anthropology. This is combining some of my favorite characters, Superman plus a little Indiana Jones. And I'll give two, hopefully quick examples. On the right, we have medical CT you see in the background, and they're putting a whole mummy in there, right? Everybody knows the first rule of mummies is you don't unwrap them because you get cursed. So you don't want to mess with it because of that, but really you don't want to mess with it because if you try to unwrap these or unravel them, they will deteriorate and fall apart. And these are so precious, we can't risk that. But we still want to figure out who the individual was inside, what they might have been buried with, how they might have died, what pathologies they might have. And this is an example of how we digitally unwrap them. The mummy is still intact, but you can digitally take off those layers and check out what's going on inside that specimen. Here's another example, a different mummy that I thought was interesting. That blue turns out is less dense material that they figured out was brain matter. And the green was like a metal rod. And they figured out that this was how they know this is how they preserve the mummies, is they remove brains and internal organs, and they put them in those little jars, and they're buried with this. But some apprentice was doing the preservation here, and he broke off the metal tip in the skull, and he couldn't remove the rest of the brain. And before his boss came back, he kind of covered everything back up and said, everything's fine, nothing happened here. But with the micro CT, or in this case, the medical CT, we can peer in and see that that guy was totally incapable of holding that position. That's right. So on the left-hand side is another example from archaeology. It looks like just a charred piece of paper, and in fact, that's what it is. This was at one time a scroll that had some sort of text on it, but it was survived through a fire, and all that's left is this charred little roll. If you try to unwrap that roll, it's just going to crumble in your fingertips. But again, micro CT to the rescue. You throw it in there. You see on the left, that's one of those slices from the Z-stack, so a cross section, and you see that scroll wrapped up on itself. And they were digitally able to unwrap that scroll. And that picture on the left is not how the specimen actually looks. That is a digital unraveling of the specimen. The specimen is still the thing on the right, that charred, wrapped up little scroll. And when they unraveled it, they noticed that there's a density difference on the paper. There was the dark paper, which means X-rays are transmitting through, but there was this light squigglies on top. And they determined that was text, because the ink used to write this text had some sort of metal or more dense material. And so we can interpret the text on the parchment. And it turns out that was a passage from Leviticus that they were able to read where it was still preserved, without ever changing the original specimen. Now I'll just give you two more examples of some really cool things that we're trying out at Duke and elsewhere, adding things to our scans to make them look even more beautiful than they started off. And this we can kind of compare to the differences between a CT and an MRI. A CT on the left, we've already covered what that does. X-rays pass through, stopped by dense things like bone. That means bone shows up really well on a CT image. That's why I keep showing you pictures of skeletons. But soft tissue, not so much, because brain matter, muscles, the X-rays just kind of pass through them and don't pick up all that fine detail. An MRI, on the other hand, you probably heard about this in a hospital, uses a different setup. It uses magnets that change the resonance in your body, and they highlight soft tissue preferentially. So you don't actually see the bone very well on an MRI, but you see the soft tissue really well. Well, we want the best of both worlds. So what some researchers up in New York have figured out is that they can add iodine to specimens, and those specimens soak up the iodine into that soft tissue. Iodine is radio opaque, or it's more dense. So now when the X-rays pass through, they have a little more to bounce around in and pick up the detail. They did this with a boa constrictor head, and we're slicing now from the bottom to the top of the skull, going through scales first, and there are those muscle groups. They're now no longer just gray blobs. You can see individual muscle fibers coming out in the boa constrictor's head. In the back, you see the spine. On the right, on the left, the eyes are popping up, those big white orbs, because the lenses absorb iodine preferentially. In the front, which is the left, you see areas where the nose is, and the jaws are just starting to form there. Again, beautiful muscle groups all the way through. Not something you would have seen with a normal CT scan. But the combination of micro CT and this new staining with contrast gives us these beautiful videos. The researchers up in New York that did this won a little video award for science video because this is just amazingly beautiful to watch. So we at Duke tried to copy them a little bit. We had a researcher from Johns Hopkins, Heather Christensen, who came in with these tree shrews, also from Southeast Asia, also pretty rare and hard to come by. So the museums that have those don't just want to lend them out to researchers to cut open and destroy. She, however, wanted to look at the muscles of chewing in this animal because this is a good model for the early evolution of primates. They think that the earliest primates kind of moved around the trees in a similar way, maybe looked a similar way, were a similar size. So we use these as a model. And to see those chewing muscles without dissecting it, we added the iodine, CT scanned it on the micro CT at SMIF, and created this animation. The last video was a Z-stack. We're going slice by slice. This is a 3D model that we can still slice in various directions to see all that beautiful internal anatomy, whether it's muscle groups or organ systems Now it's peeling back through the thighs, and you're about to see the tail down to the feet. So all this new data allows scientists to have a whole new range of research questions to follow and to figure out new things about the animals that they're studying. Here, looking for little details like the tongue that's sticking out of its mouth. You'll see the heart and cross-section up in its thorax right there, some of the other organs in its gut. Just really cool stuff that wasn't possible even a couple years ago. And so with that, I'm going to wrap up my portion of the show. Hopefully this kind of inspires you to see normal objects a little differently. This technology is fairly easy to come by now. SMIF is a shared materials instrumentation facility. So as you go on through high school and college, if you ever have a research project that you think could benefit from this, look us up, or there's various other universities around the nation that have facilities like this. And bring new ideas and scan some new stuff and use that X-ray superpower that is just waiting for you. So thank you for your time.
Thank you, Dr. Gladman. Now that we've learned all about micro-CT scans, it's time to begin our activity. Tonight, you will be putting your senses to the ultimate test. There are many unknown household objects wrapped in bubble wrap scattered around the cafe. So each one of you should have a bubble wrapped object in front of you. If you don't, you can join another table who does have one. Feel free to pick them up and touch them like many of you are doing. Your table will collaborate to guess what your table's object is. On the screens around the cafe, we will be showing some cross-sectional CT images, also called Z-stacks of the objects, to give you some additional clues. Online audience, if you don't have any physical objects, you can still use these cross-sectional videos to guess what the object is. Have fun.
All right, I think everybody, do you guys have some good guesses as to what your object is? Raise your hand if you think you know what it is. Raise your hand if you have absolutely no idea what it is. Okay, so some good guesses out there. All right, well, Justin is about to reveal what these objects are. Are we ready? All right.
All right, so object one, what were the guesses for object one? Just shout them out, go ahead. Oh my gosh, you guys are too good at this. All right, so there we have the projections of the flashlight spinning around. You can actually see the foam still wrapped around the flashlight there. Very good. Okay, object one, pretty straightforward. What about object two? Apple, orange fruit, roundy fruit. What? Orange or grapefruit. All right. I hear a lot of oranges.
Lime.
So here's a 3D reconstruction. You might have had little orange wedges when you were much younger in school.
Ew. It's kind of gross.
And we'll see that transform into the full orange here. Okay, so orange was right. A lot of right answers once again. Okay, what about object three? Anybody got a guess for object three? A lock? A lock? I have a lot of consensus on a lock. So next question, what is the combination to the lock? I don't know, I locked it a long time ago. But realistically, you could now pick this lock by peering inside and looking at where those different pin positions are. And in fact, I tried to do this. Turns out it's harder than it looks. You not only have to be able to CT a lock, but you have to know something about how the lock works. So I'll get back to you when I actually figure out what the combination is. And object four, oop, I went too quick. Object four, TV remote, anything else? Somebody's one for one then. This is in fact a TV remote that's spinning around. And here you just see the circuitry, and then it'll build up the outer shell, which is a less dense plastic. Very good. So it turns out most people here don't need to use the micro CT because they have perfect X-ray vision. But if you have something a little more complex or you're interested to see what the inside of these specimens look like, micro CT is just perfect for that.
Cold.
Can we switch sides? Can you have your normal side? Can I have a side? Yeah.
Okay, the floor is now open to any questions you may have for our speaker and the research. We will have 10 minutes to questions, so if we do not get to your question, you can come up afterwards to ask it after the presentation is ended. To those who are watching on Facebook Live, you may ask questions in the comments section. We are monitoring the comments to voice these questions for the speaker.
Any questions? Yes.
Does the CT scan affect like the technological equipment that you put in?
No, it shouldn't affect anything that's already dead. So TV remotes and cell phones should be perfectly fine on the other end, but I wouldn't put any of your friends in there because it wouldn't go so well. It's like a medical CT. We use a similar radiation, but much longer exposures. So, you know, everybody knows that X-rays can be quite damaging. If something living was in there, they'd be exposed to way too many X-rays and it probably wouldn't end very pretty. So we try not to put anything alive in there.
How long did it take for the ant head to print?
Where are you? Oh, sorry.
How long did it take for the ant head to print?
To print out, it took the better part of 3 days. And I did it at Duke has a shared 3D printing station that's free, but they don't expect you to actually print something on their printers for 3 days, so they weren't really happy with me when I went to pick it up.
So when you scan it, how do you capture the, like, how are the images captured when you?
So that's the X-rays pass through, and normal X-rays, like at a doctor's office, or at least 20 years ago, this would have all been done with film, right? Just like you used to have in cameras. Even that's starting to become a dated reference. But film would be exposed. You take it to a dark room and you process it to get a picture. Now we do this all digitally. So that detector you saw on the back was all digital, and it's just able to interpret the signal that it gets and create the beautiful image right on the spot. kind of like a digital camera.
How much does that equipment cost that you're using to make these scans?
That equipment, the micro CT, is more than $1 million to buy, and then the upkeep alone is quite expensive. So this is why it's in a shared facility on campus and not just stuck in one person's lab, so that everybody can come use it, and then the costs are kind of split amongst several different researchers. I think we have an online question up here.
All right, we have a question from Maggie Hall. A PET scan gives off more radiation than an X-ray. So healthcare officials try to give PET scans in order to expose patients to the least amount of radiation possible. How does micro CT compare in terms of amounts of radiation, and how safe is it for a living organism?
Well, there you go. So that probably came in right before I was talking about you don't want to put your friend in there.
Yeah.
The micro CT and the medical CT probably have overlapping amounts of radiation, but a medical CT is programmed in a way that it zaps you as short a time frame as possible so that you're exposed as little as possible. The micro CT, we don't really worry about that because we're doing it only for research purposes. So as a general rule, living things in the micro CT is a no-no, and it's probably more radiation than any of those other medical technologies.
Also, one more question. Are there any practical applications for healthcare that Duke could use?
Oh yeah, there's some great applications. So one thing we're doing is I do teach a little bit of anatomy over in the Med Center. And we're trying to take some from a gross anatomy lab dissection material and scanning it so that future med students and PA and PT students can learn from anatomy dissections digitally again, because sometimes when you do a dissection, it's kind of a one and done thing. This way we're able to pass it along to different generations of students. Also, there's various research purposes. One group scanned, I think, a pig heart and captured the valves of the heart. And then they're able to go to the 3D printing lab and print off those heart valves using a flexible material. The idea here is that you can start to generate your own biomaterials using first the micro CT and then things like 3D printing that you can ultimately implement in healthcare in the doctor's office.
So does the CT scan affect any data in the USB drive?
You know, I don't know if the USB drive had anything on it. It's possible. I don't actually know if it would affect the kind of the storage of data. In theory, no. And the reason why CTs, I said, are used for characterization or quality control is a lot of big factories have these units in them, like big cell phone factories, for example, and they'll throw every 100th cell phone in there to make sure that it looks like it should. That's quality control. But that's before they've ever sold it. So presumably it's not damaging that material at all. Otherwise, they're ruining their own product.
All right, I have one last question. So this is a really super cool line of work, but it's not very conventional, I suppose, a lot of people get into. So I was wondering, how exactly did you get into this, and what drew you to this line of work?
I'm not an engineer, even though I work in the engineering department. I started off doing some of the scans that you saw. So those little primate scans, that's the type of thing I'm interested in. My work is researching primate evolution in South America, and I decided to CT various parts of the skeleton to create these beautiful models and analyze the shapes to try to determine what they were doing and how they're related, et cetera, et cetera. And I spent so long scanning that the people in the lab just took pity on me and ultimately decided to give me a job running the machine to give me a real outlet for my time. So I kind of stumbled into the position through use of the machine and through an interesting research question that, you know, intersected with this interesting piece of technology. Thank you.
Sorry to cut questions short. If you have any other questions, again, you can come up after we're done here. Thank you so much, Dr. Gladman. Let's give him another round of applause. Who knew you could use CT scans to look at ancient scrolls, right? Thank you for coming to the cafe, everybody, and thanks to our online audience for tuning in as well. We would also like to thank the wonderful cafe staff for allowing us to use the space. Don't forget to follow Teen Science Cafe and mention us on Twitter, Facebook, and tag us on Instagram. Also, maybe there's other social media out there. I don't know. Come in March for our next cafe when we talk about the dangerous impact of floodwater. It's cooler than it sounds. I'm sure of it.
All right. So if you love science, come check out Darwin Day, Saturday, February 11th, to learn all about evolution, which is just cool. On February 26th, there will be a teen volunteer fair where you can come and learn about each program, meet current participants, and get your questions answered. There will be some purple shirts there. There'll be some junior curators there. I think we got some of them over here. Some teen newsroom. It'll be a great party. And learn about stuff. Maybe you could do it. It'd be lots of fun. We'd love to have you. There will also be an adult science cafe on February 9th. Outrageous heads led to outrageously large dinosaurs. That's the first time I've seen that today. So then another on February 16th on Alzheimer's and your heart, how exercise helps. So thank you all for coming. Woo! Science. So any additional questions, you can come up or you can look at the ant head, which is pretty awesome. And have a nice night, everybody.